pericardial, peritoneal, and pleural cavities. It also lies cavities of organs, for example, uterus or urinary bladder, blood vessels. They are all lined by epithelial tissue. And also epithelial tissue forms organs such as glands. So epithelial tissue is subdivided into main groups, covering epithelia and secretory or glandular epithelia. Today we will talk about covering epithelia. So for next lesson, study only covering epithelia until glandular. This is chapter 4 in Janquera's basic histology. You need Janquera again now. Chapter 4 is epithelial tissue, but study not till the end. Study until glands. Is it clear for you? What is your
Until
homework?
what? Until what, doctor?
What?
Until what?
Until glandular epithelia.
Okay.
Only covering until glandular. Is it clear now? Mohammed,
Okay,
chapter
clear, clear, doctor.
4 until glandular. So all tissues consists of cells and extracellular material, but amount of extracellular material in different tissues is different. In epithelial tissues, there is very small amount of ECM, and this tissue mostly consists of closely aggregated cells. You can see here closely aggregated cells with a strong adhesion, and that's why they form continuous layers. They form sheets lining body cavities, cavities of organs, and covering our body. Cells are attached to each other by different junctional complexes, and you should know for next lesson all these types of junctional complexes that connect to neighboring cells in this tissue. Uh, Shape of epithelial cells may be different. They are squamous. Some of them are cuboidal. For example, these cells, they are cuboidal. It means that they are as tall as wide. And epithelial cells may be tall, columnar. It means they are taller than they are wide. And pay attention that shape of nuclei corresponds to cell shape, because in columnar cells, nuclei are elliptic, ovoid. In cuboidal cells, they are rounded, spherical, and in flat cells, you can observe flattened nuclei. Shape of cell and number of cell layers are main criteria for classifying epithelial tissue. So let's classify epithelial tissue. It's divided into groups, simple and stratified. Simple epithelial tissue consists from one layer of cells. Look here, only one layer of cells. But shape of according the shape of cells, simple epithelia may be squamous, cuboidal, or columnar. Endothelium is simple squamous epithelial tissue because it consists of single layer of flat cells. You know that this tissue develops from angioblasts. Do you remember blood islands? When cavities appear in the blood islands, we discussed this previous lesson. Some angioblasts, they flatten and they become endothelial cells. So you can observe here endothelial cells, they line blood vessels. This is scanning electron micrograph. It's cross-section of artery, and inner layer of artery lining the lumen of this organ is endothelial tissue. So it's squamous, simple squamous epithelial tissue. Now you can observe longitudinal section of blood vessel with light microscope. So this is longitudinal section of blood capillary, and these dark stained nuclei are flattened because squamous cells contain flattened nuclei. So all cardiovascular system is lined by endothelium. Another type of simple squamous epithelium is mesothelium, and it lies peritoneal, pericardial, and pleural cavities. This is simple cuboidal epithelium. It means that there is only one layer of cells, and these cells are cuboidal and they contain spherical nuclei. You can find this tissue in the kidney, in the thyroid gland, in the ovary. So these cells are cuboidal with spherical nuclei. Another type of simple epithelia is simple columnar epithelium. It lines stomach, for example, it lines intestine. These cells are tall cells with elliptic nuclei. Pay attention that cuboidal and columnar cells show polarity. What is polarity? Uh, Only five students are now present. Why? Where is six student?
Dr. Layla had to leave the class because she have work.
So she does not need explanation. So
Uh,
it's
I
up
will,
to you. You can listen or not as you like.
of course, doctor, we want to listen.
So what is polarity? It means that two poles, two regions of the cell differ functionally and structurally. This surface adjacent to lumen is called apical surface. The opposite surface adjacent to underlying tissue is called basal surface. So these two parts of cell differ from each other. Adjacent tissue is always connective tissue. And structure that lies between epithelial tissue and connective tissue and separates these tissues from each other is called basement membrane. Can you see here this line? It's basement membrane, it's stained basement membrane, and it separates epithelial tissue from underlying connective tissue. So this surface of the cell that is attached to basement membrane is called basal surface, and this surface facing the lumen is called apical surface. Nuclei are usually located in the basal region of the cell. Rough endoplasmic reticulum is also located in the basal portion of the cytoplasm, while Golgi apparatus and secretory granules are located in the apical surface of the cell. So distribution of organelles in apical and basal part of cells is different. Uh, Sometimes at apical surface of epithelial cells there are specialized structures, for example cilia or microvilli or maybe stereocilia. You can see here now long microvilli on the apical surface of cuboidal cells. And you know that absorptive cells lining small intestine also have many uniform microvilli on apical surface to increase area for absorption. So on apical surface of epithelial cells, you can find specialized projections such as microvilli, cilia, and stereocilia. You already know the structures because we have studied it during cytology, but you should revise this. I will ask you structure of cilia, stereocilia, and microvilli next lesson. At the basal surface, Some cells have basal invaginations and they also increase surface area. Now you can see a simple columnar epithelium. So this region is apical and this surface is basal surface. Another type of simple epithelia is called pseudostratified. Why it's called pseudostratified? Because it has stratified appearance, but it's not stratified. We can find this tissue in the respiratory tract, trachea, bronchi, nasal cavity are lined by this tissue, pseudostratified epithelium. This tissue contains three different types of cells. Small cells, look here, they don't reach the surface of the tissue. Tall cells, they are ciliated. And goblet cells, they don't have cilia, they produce mucus. So these three types of cells have different morphology, size, shape, and that's why their nuclei are located at different levels. You can see here several rows of nuclei because they are not located at different levels. And this gives this tissue stratified appearance. But this tissue consists of only one layer of cells, and all these cells are attached to the basement membrane. That's why it belongs to the simple epithelium. Now all types of simple epithelia are shown here. I will list them once again. Simple squamous, simple cuboidal, simple columnar, and pseudostratified. These are types of simple epithelium. And all these tissues contain only one layer of cells, and all these cells are attached to the basement membrane. And you can see here basement membrane between these two tissues, epithelial tissue and connective tissue. Pay attention that epithelial tissue is avascular. It means that it does not contain blood vessels. Even thick epithelia do not contain blood vessels. Blood vessels are always located in the underlying connective tissue. So, nutrients, different substances must cross basement membrane to enter epithelial tissue. That's why basement membrane serves as a filter for these substances. So it's not only physically separates these two tissues, but it also serves as a filter. And it also has another important functions, and you will study this for next lesson. Let's classify another group, which is called stratified. I will change the presentation. So it's easy to understand that stratified epithelial tissue contains several layers of cells. For example, look at this drawing. This tissue consists of many layers of cells, and of course only deepest layer is connected to basement membrane and not other layers. There are several types of stratified epithelium. This is stratified squamous keratinized epithelium, and it is found in epidermis of the skin. What is main function of this tissue? Protection. Of course, protection because it protects our body, our underlying tissues from water loss, dehydration. And it protects against invasion by microorganisms. That's why it's thick and it's keratinized. So it consists of many layers of cells, and cells in these layers differ from each other. The deepest layer consists of cuboidal or columnar cells. You can see that they contain nuclei. They contain organelles. They are active metabolically. They can divide mitotically. But then these cells undergo differentiation. And during differentiation, they change their shape, they become flat. They lose their organelles and nuclei, and they become filled with keratin. So, in the superficial layers, you can observe squamous. They lack nuclei, they lack organelles, and they are filled with keratin intermediate filaments. That's why this tissue is called keratinized. This is stratified, squamous, keratinized epithelium. It covers our body and from it protects against microorganisms and dehydration. Another type is called stratified squamous non-keratinized. It means that it contains less keratin. Of course, these cells also contain intermediate filaments. All epithelial cells contain keratin intermediate filaments, but amount of keratin in this tissue is much more less than in keratinized. It's quite thick, you can see that it's thick because it consists of many layers. And all these cells contain nuclei and organelles and are metabolically active. This tissue also has protective function, but it lines wet cavities where water loss is not a problem. For example, it lines mouth, esophagus, wet cavities. Now another type. It's called transitional epithelium. It's also called urothelium because it lines urinary bladder and other parts of urinary system. It consists of several layers. Pay attention that superficial cells. They are large cells. They are dome-like cells. Sometimes they are called umbrella cells. And they have unique morphological features because they can change their shape and allow distension of this tissue as urinary bladder fills with urine. So you can observe now two different states of this tissue, relaxed state and distant state. When you can observe relaxed state, pay attention that superficial cells are large, dome-like cells, umbrella cells. But after distension, they change their shape and they are flat now. Look here. So these cells can change their shape and allow distension of the wall of the urinary bladder when it fills. And this tissue protects underlying tissues from toxic effect of the urine. Stratified cuboidal and stratified columnar epithelium are rare types. You can find them in some ducts of glands, for example, sweat or sweat glands or other submucous glands. So you can see here two layers of cuboidal cells. So this is stratified cuboidal epithelium. Now all types of stratified epithelium are given here. Stratified keratinized, stratified non-keratinized, transitional epithelium or urothelium, and stratified cuboidal and column. And even thick epithelia do not contain blood vessels. Blood vessels are located in the underlying connective tissue, so nutrients enter epithelial tissue from underlying connective tissue. And structure that separates these two tissues and serves as a filter is called basement membrane. So as I told you, all these tissues consist of closely aggregated cells, and they are linked to each other by different junctional complexes. You can see here tall epithelial cell. It has apical surface, as you already know. It has basal surface, and it has also lateral surface. With lateral surface, cells contact each other, neighboring cells contact each other. And at the lateral surface, the cells have different junctional complexes. Most apical junction is zonula occludens, tight junction or zonula occludens. And you can see that membrane of neighboring cells are fused in several points. So this is one cell, this is another cell, and membranes are fused. That's why this junction forms a seal between two neighboring cells, and it prevents passive flow of material between cells. So substances move through epithelial cells rather than between them, because zonula occludens forms the seal between them. And this junction is called zonula because it's like a belt. It surrounds entire cells. It encircles the cell. Immediately below zonula occludens, you can observe zonula adherens. This is zonula adherens. And in this region, membranes are not fused. This is adherent junction. It holds cells together, but fusion does not occur. It's also called zonula because it's also belt-like junction encircling entire cell. And zonula adherens stabilizes zonula occludens. Next type of junction is desmosome. Look here, it's also called macula adherens because unlike two other junctions, it's not belt-like, it's spot-like junction. It consists of two parts. One belongs to one cell, another belongs to another cell, and transmembrane proteins, which are catenanes, adhere cells to each other. So proteins of catenane family are components of desmosomes. And pay attention that cytoplasmic filaments, which are intermediate filaments, are attached to the desmosomes. Keratin filaments are very strong filaments. That's why desmosomes form sites of strong adhesion between cells, and they maintain integrity of the tissue. So desmosomes are spot-like junctions, and they are strong sites of adhesion between cells, and they maintain integrity of the tissue. So you can see here desmosomes with electron microscope. So several junctional complexes are visible here at the lateral surface of the cell. as I told you, most apical is zonula occludens. Just below it is zonula adherens, and then you can see several desmosomes. So this is lateral surface. This is apical surface. You can see microvilli on the apical surface of the cell. Another type of junction is gap junction, and it differs from previous junctions because it is communicating junction. So function of gap junction is communication rather than occludence or adhesion. So you can see here membrane to membrane of neighboring cells. They are very close to each other. There is very small space between them, only 3 nanometers, but membrane are not fused. And each membrane contains cylindrical structure, which is called connexon. And connexons are paired. You can see paired connexons. And each this cylindrical structure or connexon has channel inside. That's why small molecules can pass from one cell to another cell. Diameter of this channel is approximately 1.5 nanometers. That's why molecules that are smaller in diameter than 1.5 nanometer, they can pass through the channel. So ions and other small molecules are transported between the cells via gap junctions. This is communicating junction. And this junction is found not only in epithelial tissue, but it has very important role in muscle tissue and in nerve tissue as well. Now, which junction connects epithelial cell to basement membrane? You know that basal surface of the epithelial cells is connected to the basement membrane. And junction which connects basal surface to basement membrane is called hemidesmosome. It is called hemidesmosome because it resembles a half of desmosome, but unlike desmosomes, they contain integrins rather than catenanes. So they contain different family of proteins. So now you know everything about epithelial tissue. You know types of epithelia. You know specializations at apical surface of epithelial cells. You know junctional complexes between epithelial cells. So this is your homework for next lesson.
Doctor, can I ask
What?
something? Can I ask something?
Yes, of course.
You said we have chapter 4 until what title?
Until glandular epithelium.
Okay.
Only covering until glandular. Is it clear now?
Okay, doctor.
Do you have any questions?
No, thank you, doctor.
No.
Thank you, doctor.
We can finish our class today.
Goodbye, doctor.
See you next week. Study to prepare this material very well, okay?
Okay, thank you.
Thank you so much, doctor.
So bye. See you next week.